Go.
Welcome observers, my name is Gina and this is Harsheen.
Hello. We're here to learn about three processes that go on in a genome.
We are currently in a nucleus inside of a cell. Harsheen, what is a cell?
The cell is one of a billion components that make up organisms.
This is a model of DNA, deoxyribonucleic acid, the double helix molecule that contains all the genetic information of an organism.
DNA is a polymer made up from many nucleotides. A DNA nucleotide is made up of a phosphate group, a deoxyribose sugar, and a nitrogenous base. See those blue parts? Those are the deoxyribose sugar groups of a nucleotide.
One end of one strand on the right side has a 5' end, and the other end of the strand is known as the 3' end. The other strand on the left side is anti-parallel to the right side strand, meaning that the 5' end is not parallel to the 5' end of the other strand. And the same thing goes for the 3' ends. As you can see, the 4 DNA bases go with a relationship called complementary base pairing. For this, the base guanine, which is represented by the color yellow, is a complementary base pair of cytosine, which is represented by the color orange. And the base thymine, which is represented by purple, is a complementary base pair of adenine, which is green in this DNA structure. If you look closely enough, you can see that it takes 3 bonds, specifically called hydrogen bonds, which are represented by the toothpicks, to connect guanine and cytosine, and it takes 2 hydrogen bonds to connect thymine and adenine. Those tacks represent the phosphate groups of a nucleotide.
DNA makes you what you are. But how does DNA do that? Well, when a cell needs to divide, more DNA is needed. So the DNA goes through a process called replication. The double helix DNA strand unravels by the enzyme helicase.
The helicase starts from one point on the DNA strand and starts breaking the hydrogen bonds between the complementary bond pairs, one at a time, eventually creating 2 strands. The original strands that are used as the templates later on are known as the parent strands. Once the 2 strands are separated, free nucleotides from the surrounding nucleoplasm bond with the unpaired bases on each of the parent strands, creating 2 new DNA double helix strands, also known as daughter strands. This bonding process occurs in a very particular way. DNA polymerase enzymes help catalyze the bonding between the 2 bases to create hydrogen bonds. The nucleotides begin bonding from one end of the parent strand and make its way upstream of the strand, meaning it is going towards the 3' end of that strand with the help of the DNA polymerase. Once the strand is done bonding, this enzyme goes upstream of the other strand to the 3' end. Notice how these DNA double helixes are semi-conservative, meaning both new strands have one parent strand that came from the original DNA molecule. So we know one parent strand is always passed on to the new DNA double helix. As you can see here, the sequence of base pairs are identical. Another process that is taking place here in the cell is protein synthesis. Here, the first set of reactions taking place is transcription. During transcription, a section of DNA called a gene produces messenger RNA, a smaller section of RNA that carries information for protein synthesis outside of the nucleus. A DNA gene begins unraveling. There are free RNA nucleotides in the surrounding nucleoplasm that begin bonding using one of the 2 DNA strands as a template. A RNA polymerase enzyme helps catalyze this bonding to occur. A RNA nucleotide is made up of a phosphate group, a ribose sugar, and a nitrogenous base. The 4 bases of RNA nucleotides are adenine that bonds with uracil and guanine that bonds with cytosine. Note only one DNA strand is used because the RNA molecule is a single strand molecule. This is the antisense strand. See how the mRNA produced from the antisense strand contains the same sequence as the sense strand. Also, see how the mRNA strand that is copied is shorter than the DNA strand. This mRNA strand has enough information from the DNA strand to create one polypeptide, a linear sequence of amino acids. And the information in an mRNA strand contains the codons, which are specific sequences of 3 nitrogenous bases that code for a certain amino acid to determine a specific order for the amino acid chain, a polypeptide.
The second part of protein synthesis is translation. After the mRNA is finished with the transcription, it exits the nucleus through nuclear pores and finds a ribosome and begins aligning itself specifically. Then, a tRNA transfer RNA with a specific anticodon that corresponds with the mRNA's codon joins, bringing along a specific amino acid. A second tRNA brings a specific amino acid and ends up beside the first amino acid. An enzyme catalyzes a condensation reaction that joins the 2 amino acids in a covalent bond called a peptide bond. The first tRNA has served its purpose and therefore it breaks its bonds with its amino acid and exits to reload with another amino acid. The second tRNA moves over one spot and makes room for a third tRNA to arrive with another amino acid. This process continues to form a polypeptide chain until the last codon of the mRNA gives the code stop to the polypeptide. After translation is finished, the polypeptide breaks away from the tRNA and is free in the cytoplasm of the cell, free to do whatever it is synthesized to do. Well, that is all for today's journey into the cell. Hope you enjoyed yourself and learned a little something. Until next time, goodbye.